Hey Golden Flashes, welcome to another episode of What's the Big Idea? We're on our way to meet Dr. Lique Coolen from the Brain Health Research Institute and to talk to her about her fantastic research on the brain and brain health. It's Dr. Lique Coolen. Welcome
Hello,
to the
President
What's the Big Idea, Dr. Coolen. Good to see you.
Good to
Come
see
on
you.
in.
Thank you.
Thanks for joining us for
Oh,
this
it's
episode.
my pleasure.
Welcome to What's the Big Idea?
Terrific.
Could you just tell us a little bit about your research?
Yeah, absolutely. The central nervous system is the most fascinating part of our entire body. It controls everything that we do, not only how we think and what we think, but it controls, in fact, every organ in our body as well. And so it's complex. And that makes it fascinating because there's so much to discover. It's also fascinating because there are so many things that can go wrong.
So injuries is an obvious one.
Injury is an obvious
But
one.
what else? Are there, I don't know, viruses or something?
Yeah,
that can cause
viruses,
trouble?
but also sometimes, things don't connect that well or different, I should say, leading to differences. All of our individual differences are all based in how our central nervous system differs. And that is really fascinating to study.
Dr. Coolen, I know you have some students in here doing their work right now, so why don't we go in and you can introduce me to your students, and what are you working on?
I am currently preparing PB for Rachel's experiment. So she's going to dissolve antibodies in the solution.
So, keeping in mind that I'm a historian, what is PB?
It's a buffer or a solution that we dissolve things like antibodies in.
Oh, okay. So you've got ramen noodle here and it's heating up.
Yes.
No, okay, not really. What's going on here?
So right now we are doing a procedure called immunohistochemistry. It's actually a follow-up study that Logan and I and Tywell worked on. We're currently trying to look at dopamine 3 receptor antibody and how it innervates and communicates with the LST neurons by also probing for galanin.
Can you just tell me a little more about some of the research that's being done in your lab, spinal cord, and maybe there's an injury or maybe through age, and then how do you restore functions in men or women, be it reproductive, be it other? Is that a fair description of what you're doing?
Absolutely, yes, and it's a great question. So we're doing this in multiple steps. First, we try to find out how the spinal cord normally functions in a healthy situation, what the molecules are that the neurons are using to communicate with each other, like those are called neuropeptides or neurotransmitters. Then we try to find out when there's a trauma, what is now different.
So here we've labeled the cells which are responsible for controlling reproductive function in a rat. This is actually in 3 dimensions. We're looking at a rat spinal cord. It goes in there. And so I'm interested in studying how these cells are connected both with each other and with other cells which regulate male reproductive behavior, and ultimately how these connections are altered by spinal cord injury so that we can try to restore this network function to its proper configuration.
This is called a light sheet microscope. And what this microscope can do is it uses light sheets, which are lasers that go horizontal, scan the entire tissue. And what that allows us to do is to look at an entire brain all at once, or an entire spinal cord all at once.
And this image here would have come from this microscope?
That's right.
Is that right?
Yeah.
Wow.
And we're actually looking at several inches of spinal cord all at once, which is not possible with anything but this technique.
Wow.
This is one of the few microscopes of its kind here in the state of Ohio. So Burgundy did another technique that we use to visualize messenger RNA in cells. So looking at gene expression that may be altered by trauma. And she's currently analyzing some of our data, and we're actually writing this up in a publication that Burgundy will co-author. So this is a technique that we can use to look at every puncta is actually one messenger RNA in the cell, and we're asking simply, does trauma have a big impact on that gene expression? And then what can we do to restore that gene expression? Because we know that those genes and translating to the proteins are crucial for the reproductive function that we study.
So when you ask the question, does trauma have an impact or an effect, I mean, how close are you to answering that question?
For some of those questions, we are pretty close to answering, and we already have some very promising data in our preclinical models. Now the challenge is to bring this to patients.
All right, so Dr. Coolen, thanks for joining What's the Big Idea? So exciting to listen to your descriptions of the research, the impact it'll have in improving people's lives and the quality of life. And also, thank you so much for being such a great mentor to our students from the PhD level to sophomore in college down to high school students that work in your lab. That's very impressive. That's really what Kent State's about.
Absolutely.
Thanks for joining us.
Thank you.